Welcome back, students. Today we are going to discuss part 4 of Unit 2, that is Unit of Life. So, before we start the main lecture, I would like to inform that the images used in our slides are for education purpose only. We respect intellectual property right of the original creator and owner of these images. If you are the owner of any of the images used in this PPT or have any concern, any issue, please contact us at sathee@iitk.ac.in and we shall remove the images. And in today's video lecture, we are going to discuss allosteric modulation, irreversible activation of enzymes, right? So, in the last lecture, we have discussed a lot about enzymes. Okay, so in the continuation of that lecture, we are going to discuss these two things and some kind of graph also we are going to discuss in this particular video lecture. So, let's start. As you know, that enzymes are the biological catalyst. Okay, these are the biological catalyst, means they actually speeden up a particular type of reaction. Okay, so normally these are different with the chemical catalyst in a way they are not able to work on high temperature or if we have altered the pH, they only work on the in a particular optimum range of temperature, generally 30 to 40 degrees centigrade, right? So these enzymes are biological catalyst, and this enzyme term has been given by Koehner and first discovered by Louis Pasteur. This already we have discussed in last video lecture. What is the catalyst? This catalyst is defined as a substrate or substance that increase the rate of reaction. Okay, without being itself change in the process. Similarly, enzyme doesn't start a reaction, doesn't stop a reaction. Enzyme is not change the energy level of a reaction, but yes, reduce the activation energy. So quickly, what will happen? That particular reaction start and speeded up. How? By increasing the rate of reaction. This all we have discussed in previous lecture. Okay, and we have also discussed some factors, right? How temperature affect the enzyme performance, how pH affect the enzyme performance. As for just a quick revision, I just draw a graph over here for you. This kind of graph we have achieved, right? And this is the optimum limit. If temperature is there on the x-axis and rate of reaction over here, rate of reaction. Okay, so this is the optimum range. And after optimum range, then what will happen? Again, we proceed the reaction. Then what will happen? The activity of enzyme has been going down, right? And why? Because of denaturation. Because at high temperature, enzyme is not able to perform its work, right? That's why it is written over here. The reaction rate increase with temperature initially till optimum temperature. Okay, optimum around 40 degrees centigrade. But at higher temperature, the rate decrease due to enzyme denaturation, right? Similarly for pH. pH value determine ionization of group in active site and its surrounding. So constant pH of our body fluid is maintained by buffer system. Each enzyme has its own optimum pH and that work on that particular pH only. So similar kind of graph we also get for this pH also. This kind of graph, same type of graph. Okay, after optimum, if pH is there on the x-axis and rate of reaction over here, then after optimum, okay, each enzyme work on a particular type of pH, means our stomach is having a highly acidic environment, so acidic pH. Buccal cavity is having another type of pH. And similarly, for all the biochemical activity, we need all the intracellular enzyme is able to perform their work on their particular pH. After this particular optimum situation, if this is the optimum pH, say for example, this one is 7.2, for example, or 7.4 slightly. So after that, again 7.5, 8, 8.5, the activity has been decreased. Okay, so this is a both type of, and also before that enzyme, if the medium is slightly acidic, highly acidic or alkaline, this one is slightly alkaline, right? Highly alkaline situation or highly acidic situation, in both the situation enzyme will not perform. Clear? So these two parameters we have discussed. Then after that, activators. Okay, activator means some metal ion act as a activator, like calcium ion, magnesium ion, zinc, manganese. These actually act as a cofactor, bind to the enzyme and proceed the reaction very fast. Okay. Similarly, some kind of inhibitor. This we will be discussing in this video lecture in a very detailed way. And similarly, this substrate concentration. Right. So what is written over here in inhibitor? These inhibitor actually slow down or stop the enzyme reaction. Why? Because their structure is, they mimic the structure of substrate and just go and bind to the active site of the enzyme. This enzyme is having, if this is a kind of enzyme, for example, and this is a substrate type, and this is a inhibitor, these are structurally similar, so they can mimic, they 100%, their shape type is similar to the substrate type, and if the concentration is comparatively more in that particular reaction, then what will happen? Inhibitor come and bind to this particular active site, so they actually inhibit the reaction by binding instead of substrate, that particular active site. Okay, so they compete for the active site with substrate, like certain heavy metal ion. These are strongly bind to important enzyme. Now, substrate concentration. At high substrate concentration, enzyme is saturated. Okay, so the reaction proceed with maximum velocity and graphically expressed as saturation curve. This particular last point we will be discussing in detail in the coming slide. Okay, so these are the factors which affect the enzymatic reaction. So let's discuss substrate concentration, and this type of graph is known as saturation curve. Saturation curve. Okay, and this type of graph is known as, perpendicular hyperbola. What is the name for this? Sorry, rectangular hyperbola. Rectangular. Can I write it down? Rectangular hyperbola. Polar type of graph is this, and popularly known as saturation curve. Then just see the graph. On the x-axis, there is a concentration, substrate concentration, and on the y-axis, rate of reaction is there, or we can say velocity of the reaction velocity. Okay, initially, as we increase the concentration of substrate, the reaction rate increase very fast, almost linear, in a very linear way. After some time, if we continuously increase the substrate concentration, after achieving a point, and at this point, enzyme gets fully saturated by the enzyme, fully saturated by the substrate, means all the active sites which are present in that particular enzyme get associated with the substrate. Okay, at this time, maximum velocity has been attained by the enzyme. Okay, see, for example, there is a test tube, and these are certain kind of enzymes are there. Okay, these some enzyme molecules are there, and Okay, I have put it some, substrate continuously in the enzyme. Initially, what will happen? These all substrate come and bind to the active site. Okay, bind. When all the active site has been occupied by the substrate, this brown one is the substrate, and again continuously I am going to increase the substrate concentration. Then what will happen? Up at a specific, after some specific time, when all the enzyme already engaged or busy in binding the different kind of substrate, that particular substrate, that after that, again if we continuously increasing the substrate, reaction will not increase its rate at this time. Why? Because enzyme gets saturated by the substrate. Okay, at this point we can say the reaction velocity at its maximum, at its maximum. Okay, so just read, I'm going to read these points first for you. Like the reaction velocity or V0, which we have taken on the y axis, rate of reaction or reaction velocity will increase rapidly, initially, will increase rapidly at low substrate concentration over here, as you can see, right, at this point, and then level off to a flat plateau at this flat plateau at higher substrate concentration. Why? Because all the active sites of all the enzyme is busy. Okay, and this plateau occur because enzyme is saturated, means all the available enzyme molecule are already tied up in processing substrate. This is the reason. Okay, that's why over here this type of rectangular hyperbola we will get. Okay, if at this particular point of time, if we again adding substrate, continuously adding substrate, or any additional substrate molecule will be, simply have to wait around until another enzyme become available. So the rate of reaction is limited by the concentration of enzyme. That's why over here stationary phase is there. Another type of substrate molecule we have to wait. At what time? The enzyme get free from the previous substrate, convert that substrate into the product. Okay, so this is the relationship between concentration of substrate and enzyme activity. Okay, so concentration of enzyme act as a limiting factor. Okay, only limiting enzyme is there. If you have put it 10 times substrate, then there is no need. Enzyme only bind that concentration of enzyme, means for that its active site is there. Okay, now let's move to the next slide. And this, at this point of time, what I have written over here, Vmax, or we can say maximum velocity, or Vmax, means this is the maximum rate of reaction at this particular point of time, is characteristic of a particular enzyme at a particular concentration, and it is, we can say, the maximum velocity. This is the maximum velocity of an enzyme. Okay, so Vmax is the maximum velocity. Now there is another term I have written over here on the x-axis, that is Km. What is this Km? This Km is a constant of or we can say substrate concentration, which give half of the maximum velocity. Means at this particular concentration of substrate, at this particular point, half of the substrate, half of the enzymes, not substrate, enzyme gets saturated. As you can see, at this particular point, half of the enzyme get saturated, and this point is Vmax by 2. This is just half of this whole data at this particular point. V by 2 means half of the enzyme gets saturated at this particular. Okay, when substrate concentration is this, right? And this Km is the that substrate concentration, and in Michael-Menten constant, this one is known as Michaelis constant. Okay, so this Km is very useful to measure how quickly reaction rate increase with substrate concentration. Means if we achieve low level of Km or lower the value of Km, it will show if Km is, I'll put Km over here. This is my Km value. Then what I can say? I can say this substrate is having high affinity for enzyme. Okay, if this one is in any other graph, if any other graph I can, if my Km is present over here, then what can I say? I can say that this is not the exact graph. Okay, over here, I think you understand what I want to say. That if lower the value of Km, it shows high affinity for the substrate. If higher the value of Km, okay, it means lower the affinity of substrate. So this Km is a measure of enzyme affinity towards the substrate. Okay, so that's why it is very, very useful. At this particular concentration of substrate, half of the velocity of this enzyme has been achieved. Okay, now let's discuss some classification and nomenclature of enzyme. So according to International Union of Biochemists, enzymes are divided into 6 functional classes and are classified based on their type of reaction they catalyze. Okay, hydrolases, oxidoreductase, lyase, transferase, ligase, and isomerases. These are the type of enzymes. So oxidoreductase class, all the enzymes belong to this particular class have been involved in transferring of electrons. Means oxidation reduction reaction. In transferases, transfer of group occur. In hydrolases, hydrolytic reaction occur, means transfer of functional group to water. Lyase, breaking of various chemical bond by means other than hydrolysis and oxidation. Other than hydrolysis and oxidation. You must remember. And forming new double bond or new ring structure. Lyase. Isomerases, transfer of group within molecule to yield its isomeric form. Okay, ligases, they actually joining, if we want to join two different groups. Okay, so joining of two molecules establish a new chemical bond, then we have to use this ligase. So these are the reaction type of their function, and this is the list of different classes of enzymes. Okay, now chemical composition of enzymes. All enzymes are protein, you know this very well, but there are some exceptions like some nucleotides like RNA molecule, ribonucleases. They can also act as an enzyme. But broadly, we can classify enzyme into two different categories. Some are in a very simple proteinaceous form, some in a conjugative proteinaceous. So if conjugated protein act as an enzyme or any enzyme which are in a conjugated form, that particular enzyme is having two different parts. One part is known as apoenzyme, another part is known as cofactor. Okay, and collectively when these two parts join together, then we can use the term holoenzyme for the whole, and this is the active form of enzyme. And at this particular form only enzyme get activated and show its attraction, or now it is ready to bind with a substrate, as you can see in this particular picture. So this holoenzyme is made up of or having two parts, apoenzyme and coenzyme. Collectively, apoenzyme and coenzyme, when combined together, then this whole unit act as a holoenzyme, which is a active form. Okay, see, so this cofactor we can further divide into two different categories. If coenzyme act as a cofactor, or if any prosthetic group act as a cofactor. If coenzyme is there at this particular place, then this coenzyme actually loosely bound to the enzyme, non-covalent bond. So this one is temporary arrangement is there. Okay, while this prosthetic group, if they bind to the this apoenzyme, they are very tightly bound to the apoenzyme, or even covalent bound to the enzyme. So these are highly, permanent type of structure. Now, these are certain examples of coenzyme and cofactor, like some different kind of, vitamins, vitamin B complex, all the type of B1, B2, B12, they act as a coenzyme like thiamine, riboflavin, niacin, pyridoxine, folic acid for various type of and their part like TPP, FDA, FMN, NAD, NADP, coenzyme A. Okay, so these are the part of these vitamins, right? And they participate in various type of enzymes activity. They are the very, very important part of these enzymes like pyruvate dehydrogenase, succinate dehydrogenase, malate dehydrogenase, transaminase, one carbon metabolism, pyruvate dehydrogenase. Okay, while some other enzymes like these are the cofactors like copper is there, zinc is there, copper, iron, magnesium, selenium, manganese, molybdenum for different type of enzymes like ferredoxin, carbonic anhydrase, cytochrome oxidases, hexokinases, glutathione peroxidases, arginases, and xanthine oxidase. These are certain examples of coenzyme and cofactor. Okay, so this is all about the structure point of view. Now let's discuss some points that is related to the inhibitor. Inhibitors, certain molecules act as an inhibitor with enzyme. So in a normal reaction, what will happen? As already we have discussed that enzyme is having an active site. But enzyme is having not a single active site. Another site is also there that is known as allosteric site. Okay. So anyways, in a normal way where substrate come and bind, or we can say catalytic site is active site, right? So normally substrate come and bind to the active site. Okay. When it bind to the active site, and after the binding, enzyme get activated and show its role, breaking down of the previous bond and creating new bonds, or convert substrate into the product, right? In a normal situation. But there are certain kind of inhibitors also present. One is under this category, competitive inhibitor. Another one is non-competitive inhibitor. And third one is allosteric inhibitor. So one by one we will discuss all three. So competitive inhibitor actually mimic substrate, means they have structurally very, very similar to the substrate, and that's why they compete for the active site. They compete with the substrate for the active site. If their concentration is high, they try to bind over here, and substrate is not going to bind over here. And if inhibitor come and bind to the active site, then it will get deactivated the enzyme. At this particular time, enzyme get in inactive form, right? Like there is an example of TCA cycle, inhibition of succinate dehydrogenase by malonate in TCA cycle. This is a example of competitive inhibition. Malonate bind instead of succinate dehydrogenase. It is a ATP generation cycle. It occur in mitochondria, right? And why it happens? To control the, in some bacteria, like there are certain pathogens are there, like bacteriophages are there, and other things which come and kill the bacteria. So bacteria also develop a mechanism to reduce their activity, their pathogen. They actually uh, do this type of competitive inhibition. they bind their a type of molecule, so their enzyme get inactivated. Enzyme get inactivated, that further their progeny will not develop inside a bacterial cell. That's how. Okay, so this is the example. Inhibition of succinate dehydrogenase by balonate is a example for competitive inhibition. Okay, so but this binding is, already I told you, temporary. Is this binding is temporary. So if the concentration of this substrate get high, if we increase the concentration of substrate, then automatically this inhibitor lose or lose itself from the active site, and then again substrate now able to bind this particular location or active site. Okay. Second type is non-competitive inhibitor, in which inhibitor bind to an allosteric site. Already I told you there are so many sites are present in the enzymes. This site is known as active site, where substrate get bind normally. Another site is also present over here, which is known as allosteric. Okay, so inhibitor will not directly compete with the substrate for this particular active site. Okay, while this inhibitor bind to another site which is known as allosteric site and causing shape change in the enzyme. After the binding of this inhibitor on this particular location, it will alter the shape of this active site. So what will happen? Substrate now not able to bind over here, not able to fit in this particular active site. As you can see, this shape is different. Shape is like this, bell shape, while substrate size is circular. Okay, and that's why we have discussed na induced fit theory. Substrate, when substrate come towards the what this enzyme, that time only it change or it alter or it induce shape change in the active site of the enzymes, right? So that's how this non-competitive inhibitor also come and bind to the allosteric site, which is another site, okay, and causing inactivation of that particular enzyme. Okay, like it stop the total activity of that enzyme, because this binding is very very strong. Okay, this binding is very very strong, or we can say this binding is almost permanent, like in cyanide poisoning. Example is cyanide poisoning. Okay, this cyanide will destroy cytochrome oxidase enzyme, which is a respiratory enzyme, and very quickly the person who or anyone who consume this cyanide, within fraction of second it will get die. Okay, so this one is temporary and this one is permanent. Third type is allosteric inhibitor. In allosteric inhibitor also, what will happen? This is an enzyme and this is the active site. When the product is present, when optimum amount or enough amount of product has been generated by that particular enzyme, that time product itself act as a inhibitor, and product himself go and bind to this allosteric site and alter the shape of active site, and that's how substrate is not able to bind this particular location. And in some other situation, in some other metabolic pathway also, some regulatory molecules also regulate, like in different type of operon system. Okay, regulate their the mechanism of functionality of transcription and other regulatory pathway. How? If product is present in enough amount, then product itself act as a inducer or inhibitor, sorry, inhibitor, bind to the active site, and that time substrate is not able to bind, then that particular reaction will get stopped. Okay, now allosteric modulation. This allosteric inhibition or allosteric modulation is same. It is also known as inhibition, or we can say activation of an enzyme by a small regulatory molecule that interact at the allosteric site. Other than, already we have discussed this, right? This particular site is other than the, this is, active site, and this one is allosteric site. Okay, so this one is the active site or catalytic site, and this one is allosteric site. Okay, so these allosteric modulation or some regulatory molecule come and bind to this particular location, and then what will happen? It will change its conformation, right? So they change the enzyme conformation or shape of the enzyme. So what will happen? That time enzyme is not able to bind with the substrate, and this is the main theme of induced fit theory. This is the main theme of induced fit theory. We have discussed in previous lecture also lock and key hypothesis and induced fit theory. Lock and key hypothesis tells us that an enzyme is having a fixed shape of an active site, which is just complementary to that particular substrate. Okay, but all other researchers uh, didn't agree with this thought. But then after some time, this particular induced fit theory come, and this theory says that enzyme is having active site, but this active site create its complementary form with the substrate when substrate come towards the enzyme only. Okay, towards the enzyme only, then only it creates its shape accordingly. Okay, so the ability of enzyme to catalyze a reaction is modified. Okay, and this is the basis of so-called induced fit theory, which state that binding of a substrate or some other molecule to an enzyme cause a change in shape of the enzyme, so to enhance or to inhibit its activity only at that time when that molecule come towards the enzyme. Okay, binding of a substrate cause this. Binding of the substrate cause change in shape of, in what? Enzymes, right? Okay. Now next, the substrate, that type of substrate which actually reduce the or inhibit the enzymatic reaction is known as allosteric modulation. If they inhibit, then we can say negative modulator. If they enhance or increase the enzyme activity, that time we can say positive allosteric effector. Okay, so allosteric modulation can also occur naturally. They also occur naturally as a way regulatory biological processes such as catalysis, enzyme catalyze the gene expression. Just now we have discussed like in different operon system, signal transduction also. This is how mechanism or any metabolic pathway in a positive way or in a negative way through these type of allosteric modulation, through this type of inhibition or allosteric modulation. Okay, now irreversible inhibitor. An enzyme inhibitor is a molecule that bind to an enzyme and block its activity An irreversible inhibitor inactivate an enzyme by bonding covalently to a particular group at a active site. And okay, and this type of bonding is very strong, very you can say a very permanent type. Okay, so the inhibitor enzyme bond is so strong that the inhibition cannot be reversed by adding excess substrate, which we can do in the case of competitive inhibitor. In this case, or non-competitive inhibitor, okay, in this case irreversible inhibitor, we cannot reverse the reaction if we again try to increase the substrate in that particular reaction. that particular inhibitor cannot lose from that particular active site. That bonding so much, so strong, covalently, they bond to a particular location. There are certain examples like penicillin. Penicillin actually inhibit a bacterial growth. How? By uh, reducing the cell wall synthesis, beta lactam ring of cell wall synthesis process. How this penicillin act as an inhibitor by binding to an enzyme known as transpeptidase, which is responsible for the formation of bacterial cell wall. So this penicillin act as a irreversible inhibitor. Okay, and many drug also act as a irreversible inhibitor and just inhibit the enzymatic reactions of various viruses, bacteria, different parasites, and this is how those type of drug, antibiotic and others, reduce the concentration of virus or titer value of virus bacteria inside a body. Okay, so this is all about the reversible inhibitor. As you can see, this is the catalysis reaction. Catalysis come, enzyme substrate bind, and after converting this substrate into the product, enzyme release from its location. While if inhibition is there, then this enzyme bind to the inhibitor. So enzyme inhibitor complex has been formed, and inhibitor block the substrate. It will not, if again we increase the type of substrate in that particular reaction media, then only then also inhibitor cannot lose from its location. Okay, that's how it actually inhibit the reaction type or enzyme activity. Okay, so this is all about the topics related to enzyme. Now this particular topic has been finished. If you have any query, any doubt related to this topic, you can write down to me in comment box. We will discuss that point. Till then, take care. Bye bye.